Good evening, everyone, and welcome to Straight Science. Straight Science is an evening seminar series put on by University of Alaska Fairbanks Northwest Campus in Nome and University of Alaska Fairbanks Alaska Sea Grant, also in Nome, and you're in the home office tonight. Both UAF Alaska Sea Grant and UAF Northwest Campus, we are public servants of the Bering Strait region. We serve all peoples of the Bering Strait region, which is the homeland and waters of the Inupiaq, St. Lawrence Island Yupik, and Central Yupik peoples. Tonight, I'm already learning, because I didn't know where our chum salmon went in the winter, and I guess that one will be, that's the topic for tonight. Our chum salmon are a staple for all of us in the region, and I always say chum is king. In other parts of the state, people may shudder at that, but we really look forward to always seeing the chum salmon return. And we've been having some trouble with that in the last couple of years. And so Ed Farley's talk tonight will help give us a little more insight into what happens when the chum salmon leave the area and spend their winter elsewhere. So Ed Farley is the program manager for the NOAA's Ecosystem Monitoring and Assessment Program, and he's at the NOAA Alaska Fisheries Science Center in Juneau, Alaska. So he's a fellow Alaskan, and he's coming to us from further south. And we are so glad you're here, Ed Farley. We'll leave it at that. Take it away, and let's learn about the other part of the life we don't see of our Bering Sea, Bering Strait chum salmon. Thank you so much.
All right. Thank you, Gay, and thank you for inviting me again to Straight Science. It's been a while since I've been here, so I'm going to talk to you all about the International Year of the Salmon. It's going to be focused a little bit on the winter ecology of chum salmon in the Gulf of Alaska. One of the things I just wanted to point out is we did get some funding to analyze data from our winter surveys from the North Pacific Research Board. It's Project 2210. You can see the title there is Where Have All the Chum Salmon Gone? An Assessment of Marine Critical Periods for the Yukon River and Norton Sound Chum Salmon. I'll have more information than just what we're planning to do for that project, but I hope that you enjoy the talk. Here we go. All right, so you might be asking, what was International Year of the Salmon? So this was a sort of a start with the North Pacific Anadromous Fish Commission. That commission is an international commission. It includes Canada, Japan, Korea, Russia, and the United States. And within that commission, the scientists that take part in the meetings know through the life history of the Pacific salmon life cycle that winter is a critical period for their survival. But we don't really know that much about the winter period. It's very difficult to get out and do surveys, and we have had some surveys in the past. But this International Year of the Salmon, it was sort of envisioned in 2012. It was by a Canadian colleague, Dick Beamish, and it didn't really take legs until about 2018. And so today I'll be talking about when we finally did start the project and give you some information on some of the high seas distribution and other condition factors for chum salmon that we saw. But before I go there, I just wanted to show that there was surveys that were conducted in the past. Kate Myers and others produced a paper back in 2016 that just looked at some of the general hypothesized high seas overwintering grounds for chum salmon, and those were stocks from Russia, from Asia, from the Bering Sea and the Gulf of Alaska, and just kind of gave an overview where you can see that in the wintertime, the Gulf of Alaska does have Bering Sea stocks and some Gulf stocks. There are also, if you move further south in the Gulf, there's some stocks from the Sea of Auke, the Bering Sea and the Gulf. However, when you move into the central Bering Sea, you lose some of, while there is still the Auke Sea, Bering Sea, and Gulf of Alaska. I mean, over in the western North Pacific, it's mostly just the Sea of Auke stocks. But that's sort of the hypothesized high seas wintering grounds. And one of the things I also want to mention is that the North Pacific United Fish Commission has a website. I show it here on the slide. I really encourage everyone to go to that website. There are a number of bulletins, there's technical reports, and there are also MPAFC documents that describe a lot of what I'm going to be showing today. And then there's the Year of the Salmon website. The Year of the Salmon is done now, but they still have the website up. So if you want to go back and check out what was occurring during those years of the salmon, that's a good place to go. All right, so this is we're focused on the winter ecology for salmon. Again, this winter is a critical period for salmon in the North Pacific Ocean. And we did our surveys here in the Gulf of Alaska. I have an arrow pointing, that blue arrow. We did 2 surveys in 2019 and 2020 in that gray box that you see in the Gulf of Alaska. Those surveys occurred between February and March. And then the big survey year was 2022. And all those green line, or I should say colored lines there with the dots, those are the survey track line and the stations that were sampled by 5 vessels that were out there in the winter of 2022, February through April. You can see that our Canadian colleagues had the Sir John Franklin here in the Gulf of Alaska, left Canada, pretty much sampled this orange area. We had our NOAA ship, Bell M. Shimada, sampled this region in the green area. And then we had 2 other vessels, the RV Tinro, which is a Russian research vessel. That vessel was out at the beginning of the survey. One of the things that happened, I'm sure all of you know, is during this period, a war broke out between Russia and Ukraine. And about 3 weeks into the Tinro survey, Russia called the vessel back. And it didn't quite get as much done as we were planning to. And so we had some extra charter or extra funds left over in NPAFC, and they were able to turn around very quickly. And I can't believe how fast this occurred, but within a week, they chartered the BNN Fisheries vessel, Northwest Explorer, and we had some U.S. scientists that jumped on a plane, got over to Dutch Harbor, took off, and tried to finish what the RV Tinro was not going to do. We also had another vessel, the RV Raw Spirit. That was a Canadian charter vessel. And then they did some of the sampling here in the yellow. But one of the things I want to point out is the RV Raw Spirit used a gill net to do their sampling. So that's sampling from the surface down to about 50 feet, I think it was. And then all the rest of us, the Franklin, the Shimada, the Explorer, and the Tinro, we all used a rope trawl, which samples from the near surface down to about 100 and some feet. And what we're planning to do with all these data is try to understand the distribution migration. We have genetics. We do have otolith thermal marks that the hatcheries use. This is from the ear bones from salmon. Get some information on oceanography. We wanted to look at the winter fitness for salmon in terms of fat reserves and other metrics we might use. We're trying to get a handle on potential for competition. This is when you notice at the previous slide, we do have overlapping stocks during winter in certain locations. And what does that mean for these fish if they're potential for competition? And then one of the things that I want to note is that we were trying to get a better handle on predators. Our survey sampling gear does not sample predators for salmon. However, we're using what's called an environmental DNA. And we have some initial results from that that's kind of interesting. I'll show you. All right, so what sort of oceanographic conditions did we have out there during the 3 years that we sampled in the Gulf of Alaska in winter? So remember, we had a much more limited, these black dots show the stations that were sampled, and each year there was a much more limited survey in the Gulf in 2019 and 2020. And then here you can see all these black dots in 2022 with the expanded survey further west in the Gulf and near the central North Pacific. But the ocean conditions that we saw out there, 2019 was the end of the second marine heat wave that occurred in that region. You can see down below, these are the sea surface temperatures. This is for the months of February through April. This darker red shows that this is anomalous warming by a couple of standard deviations, which means it was much warmer out there than it was in the past in this region of the Gulf during 2019. It started to cool down in the winter of 2020 when we were out there. You can see the temperatures were cooler. And then when we get into 2022, we have real contrast between what was happening in this marine heat wave period in 19 versus 2022, where it was much cooler in the region that we were surveying. I'm only going to be able to show you data from 2019 and 2022 because in 2020, the survey was occurring in February, March, and I think most of you remember what happened then. We had COVID, and we had 3 US scientists on a Canadian vessel, and we had to attempt to try to get them off the boat and back into the US before Canada shut down the borders. This was a Canadian vessel that was coming and going from Canada. So they went about halfway through the survey, got into the island out here in Canada. The US scientists were able to get into Vancouver and make it into the United States just before they closed the border. But the US scientists were collecting a lot of samples during this year, and they had to leave all those samples on board. And these were frozen samples to look at the condition, otolith samples, you name it, genetics. And those samples remained on the ship. The Canadians actually went out, finished the survey, came back into Nanaimo. They were also in a hurry because their place was closing down. And unfortunately, they stuck the US frozen samples in an office that was not a freezer. And those samples remained there for over a year and they were not accessible anymore. Once we found out what happened, once people started going back into the office, they found those frozen samples. So more stories, in 2020 was COVID, and 2022, a war broke out. So what today I'm going to be showing you then is just data that's going to be, I have some from 2022, but most of it's from 2019 and 2022. All right, so let's get into some of the information. So this is just for chum salmon stock distribution during winter. This is the Alaska Department of Fish and Game. Sarah Gilk-Baumer did the genetic analysis. And so what we have here is the distribution. The black dot means if there's nothing there, that means we sampled there with our rope trawls, but there were no chum salmon in those locations. So the pies here are the proportion of the stocks. The stocks include the light blue, which is Japan, and then the pink, which is Russia. The dark blue are Western Alaska stocks. You can see where they got their baseline here for that. If you look at the orange, that's Gulf of Alaska and Alaska Peninsula stocks. And then the yellow is Canada and Pacific Northwest. But I just put a little circle around this region here. Remember, in 2022, we had much cooler temperatures in the Gulf of Alaska during winter, and you can see that there's a lot of distribution further south. So salmon were moving further south into where there was extra or warmer temperatures that they could inhabit that year. But Western Alaska chum pretty much encompassed this region here, which is quite a big span. Between, here's the Aleutian chain, over here on the west side, all the way over toward just near the way far south in the Gulf of Alaska, over near the Pacific Northwest in the sampling area that we were at. So you can see that the Western Alaska chum salmon do have a broad distribution during winter. So these next slides, remember, we had this broader survey area in 2022, but the data that I'm going to show you only focuses in the region that we had samples similar to what we did in 2019, just so you get a better understanding. This is not stock specific, but this is age specific for chum salmon condition. And this is winter in the Gulf of Alaska region. So that region that we had samples in both years that we could compare. And this is ocean age 1. So this would be chum salmon during their first winter at sea. And one of the things, energy density is a measure of the fat content in salmon. And one thing that's really interesting is during this, remember 19 is this anomalously warm heat wave year, as you see a real contracted energy density. And it is much lower. In fact, it's significantly lower than 2022, where we had a much more broad energy density. This indicates that chum salmon in 2019 were in very poor condition when compared to those that were caught in this cooler year in 2022. Even going off into older age groups, this is a figure that came from a Japanese colleague that has a paper in the technical report that you can find on the North Pacific Anadromous Fish Commission website. But Japan has had surveys in the Central Gulf in winter, one in 1996, they did one in 2006. And the idea here then was to compare the ocean age 2 chum salmon condition in the same region of the Gulf of Alaska where comparable samples could be taken. Now, these are older age chum. Typically, after the first year in the ocean, the older ages are more capable of handling some of the changes in their environment. But what we see, especially here with that the lengths, this is a fork length, it's the body weight, and then they can use the length and the weight and get a condition factor for the chum salmon. And what we see is that the length in those 3 years were similar. But however, when you look at the weight of these ocean age 2 chum salmon, in 2019 was much less than it was in the previous 2 years. When you take the relationship there, you see that the condition factor is much lower than for these chum salmon. Even though they're large, they're just not, they don't have as much weight. And we think that indicates that there's not as much fat in their system at this point. And they have a measure they call skinny fish, but this is the proportion or percent of skinny fish in each one of the years, and you can see 19, even for ocean age 2. What this really indicates is this marine heat wave was very stressful for chum salmon, not only for ocean age 1, but for ocean age 2, meaning that if you've got low condition, there's a chance that you're going to have a much poorer chance of survival. So if we just take a look at what these chum salmon are eating, this comes out of a paper that's going to be in a bulletin through NPAFC, it's Lori Whitecamp and others. They're all, the species are here, but I want to focus on chum salmon. And if you look at the bottom here, you can, this is the mean proportion of their diets. Their diets or prey groups are over here in the colors. But again, just trying to look at this Eastern Gulf region in 2019, 2020, and 2022, one thing that I want to point out is this green and this red, they're jellyfish. So chum salmon in 2019, in this very warm, this anomalously warm period, were eating a much larger proportion of their diet from these very poor prey items, which are cnidarians and ctenophores, which are jellyfish. If we move into 2022, when it was cooler, they were still eating some of these cnidarian jellyfish, but a bigger proportion of their diet was coming from fish and euphausiids, which are a shrimp-like prey item. And these are much better prey in terms of quality. And again, remember, we saw this real change in their energy density in terms of it being much higher during this year when they were eating higher quality prey, and much lower in this year when they were eating lower quality prey. There's another a set of scientists that are looking at the food web. This is from Troina et al. It's just, again, on the NPFC website that you can get this from one of the technical reports. This is where I pulled it. This was just looking in this winter of 2019. Orienting you here, we've got a number of different species that are occupying the water column, similar to where salmon are at, you know, during certain portions of the day. That includes zooplankton, jellyfish, squid, and myctophids. You can see over here, this is what a myctophid looks like. They're really small. And the squid species are also here, and then the salmon. So in the A, this one over on the left-hand side, it says A. This food web look here is coming from this northern section, or the northwest section of the survey grid, and then B is from the southwest. And so what we're looking at here is a carbon nitrogen ratio. Just to bring it down home, you are what you eat. And so this is a measure of looking at what part of the food web you might be eating at. And so you can move up. This is the if you move up from the lower left and keep going up until the upper right, the further you go, you move up the food web in terms of you're eating either jellyfish or you're eating zooplankton or you're eating more fish or squid. When you get to the fish and squid, that's in the upper part. But this is they were trying to get a handle on, is there potential for competition out there? And so salmon, if you just look at the circles, if you're outside, if there's some overlap in the circles, there's a potential. So you can see here, some of the salmon were outside, but then some salmon also overlap with some of the myctophid species and also some of the squid. And this is in both regions. And so the authors here are suggesting there is some potential for competition among these other species that are occupying the water column with salmon. But of course, we also know salmon eat squid. So just, again, these are the carbon nitrogen signatures. This is just looking at salmon now so salmon food webs during winter. Again, this is of how we look at the food web. It's what you eat. And if you're over in the lower left-hand corner, you're eating lower in the food web. If you're in the upper right-hand, you're eating in the upper part of the food web. And just orienting everybody here, we've got A is 2019, B is 2020. And then what the authors did is they wanted to look at the spatial differences in 2022. And since we had surveys over here in the Eastern Gulf, Central Gulf, Western Gulf, and also some in the Central North Pacific, they wanted to separate it by this FR is for the Franklin, the SH is for the Shimada, TI is Tinro, and NW is the Northwest Explorer. But what's the take-home message here? You can see that chum salmon, as I've noted before, they were eating a lot of, they do eat jellyfish and other things, but they're typically feeding at the lower end of the food web. The next one here, the sort of a brownish color, that's sockeye, and then pink is pink and coho is blue. We only caught a handful of Chinook out there, less than 10, but they went ahead and did the food web there, or did the carbon nitrogen ratios for them too. And then what you want to do is see, well, where is there any overlap among these that might suggest potential competition? And there is some, but there's quite a bit of separation here in 2019 between chum salmon all the way up to Chinook. Definitely they're feeding in the different parts of the food web. If you go into 2020, there might be some overlap between chum and sockeye, but pink and coho and Chinook seem to be feeding in the different parts of the food web. If you move here into 2022, when it was colder, it really changes depending on where you're at in the Gulf of Alaska. I think this is what they're trying to get at. So if you're in the eastern Gulf of Alaska where the Franklin surveyed, there's some overlap between chum and sockeye, and then between pink and coho. I think you can get the picture. This also occurs a little bit with the Shimada, but you see more separation as you move west in the Gulf. And then even when you get to the Northwest Explorer, there's quite a bit of separation. So what does this all mean? This is one way to try to understand, well, where might competition be occurring? Are they feeding on the same part of the food web? Are they distributed in the same places, during this time? So these are all questions that, we're still trying to get a handle on. These are all just initial papers. So they weren't really even papers, they're just analyses. But this is definitely going to be taken much further as we move down into more in-depth analysis of this. And then I just wanted to kind of give you an idea of how we might use some of these new tools. This is environmental DNA, and we're able to use the samples that we took of the water column. They run them through a machine or however they do it, and they can come up with detections. So we were looking, in this case, just detections of salmon, sharks, and Bell's porpoise, as these might be two predators of salmon in the marine environment. And this is just for 2 years, 2019 and 2020. So let me orient you here. On the top two here, it's for salmon, sharks, and on the bottom, As you go from left to right, that's for Dall's porpoise. The darker areas that you see here, that's all salmon, not just chum. So it's salmon distribution, and areas where more salmon are located are indicated by warmer or hotter spots. So the darker the red, the more salmon are there. So if you look at the relationship, and the black dots are detections of salmon sharks using eDNA. So the samples that were collected, they detected salmon sharks in these areas. And I think it's very interesting that in 2019, the salmon sharks were detected and pretty much within the areas where salmon were distributed. However, when you go over to 2020, you can see that most of the salmon were distributed quite a ways south in the survey area, yet salmon sharks don't seem to be in the region where most of the salmon were located. And then just coming down to the Dall's porpoise, based on the eDNA detections, they too were also located in the regions of salmon distribution, but not as much so in 2020. And this is just interesting to think about, where you've got potential, salmon are in much worse condition in 2019, and predators are there. So moving on to the next slide. So that's kind of the winter picture that I have for you. And I just wanted to try to connect the dots then to Western Alaska chum salmon, and try to get you oriented to remember what we saw in 2019 and 2022 for chum salmon in the winter period. And then I'm going to reorient you to what I think Jim Murphy showed you a couple months ago for juvenile chum salmon condition up here in the northern Bering Sea. Both of these periods that I'm going to show you are the critical periods for marine phase for salmon. This first summer at sea, they need to grow fast and get fat in order to help them survive winter. And then, of course, you've seen what happened in the 2019, where we had that second half of that marine heat wave, and then 2022 where conditions really changed. So let's try to see if we can connect some dots. And this is the marine migration pathways for Western Alaska chum salmon, in case that wasn't clear. I mean, they start here in the first summer, they move off into the Gulf of Alaska during winter, and then they will spend 1 or 4 more years transiting between the Gulf in winter, spring, over into the Bering Sea in summer, fall, until they start to mature, and then they move back up into the Western Alaska systems and streams to spawn. So Jim Murphy gave a talk, I believe, 2 months ago to Straight Science, kind of highlighting 20 years of data that we have from a survey we conduct up in the northern Bering Sea. It's an integrated ecosystem survey, and we have a lot of salmon information from those surveys. And one of the things that comes from those surveys, again, we started those surveys in 2002. And so I'm going to orient you here on the left-hand side. This is the northern Bering Sea sea surface temperatures. And we've had some stanzas during the time that we've done these surveys where we've had a warm stanza, which I see here is warm 1. It cooled off in the northern Bering Sea during our surveys, during this section, this stanza in the middle. And then, of course, as many of you know, it got very warm in 2017 through 19. And we have energy density data for these periods for juvenile chum salmon that we've collected in the northern Bering Sea. And during this first warm period, energy density was slightly depressed. When it got cool, we saw their energy density increase. And then during the second warm phase, we saw a dramatic decline in the energy density of juvenile chum salmon. I'm not going to go into all the details, but this does, again, fit very well with the prey that these juvenile chum salmon were feeding on. Very poor quality prey in terms of their diets in this second warm period. And again, this energy density is a measure of fitness. So it's the amount of fat prior to winter. And, many of you know, we've had 2 marine heat waves in the Gulf of Alaska. And the first one didn't seem to impact Western Alaska chum salmon that much. It did impact Gulf of Alaska stocks quite heavily. But keep in mind that during that first marine heat wave, these chum salmon in the northern Bering Sea were experiencing a cool period, much higher energy density. So they were very fit moving off into a warmer environment during winter. However, when we get to this second warm period in the northern Bering Sea and match that up in their when they leave that shelf waters and move into the Gulf of Alaska, we can see that these fish not only were in poor condition, had very low fat levels, but then they went into the Gulf of Alaska, which was also warm. And it's what we're trying to tease out here is this understanding that when we've got these extreme events occurring in multiple stages of their life histories in the marine phase, this is when we see these potential for high mortality rates. And I think this is what's happened. And so this poor condition on the shelf during juveniles off into a marine heat wave, the second marine heat wave, and high mortality. What I want to also point out is that there's some good news, I believe. Our surveys have shown that the temperatures are getting cooler lately in the northern Bering Sea. And in fact, the most recent survey data from 2022 and on indicate that their energy densities for juvenile chum have risen quite a bit. And they're some of the most energy-rich chum we've seen out there. And as we noted from the winter surveys, they've gone out into a much cooler environment now. And we note that those chum salmon are in much better shape. So I believe this is an indication that that mortality is much lower and that adult returns should start to rebound to Western Alaska. So with that, I'm going to go ahead and conclude. We know that these marine heat waves, these extreme events, have negative impacts on the condition of chum salmon, not only during their early marine residency, but also in their offshore region during winter. And that the stress likely increased overwinter mortality and could have contributed to these really poor adult returns that have occurred over the last few years to Western Alaska. Like I said, cooler conditions in 2022 are associated with improved energetic status, and really it's these extreme events that are going to pose challenge to chum salmon marine survival. And I think that when the events co-occur in life history of their migration pathways, that's when we see high mortality events. So with that, I just want to acknowledge a lot of agencies that helped us participate in this International Year of the Salmon. And I think I'll end it there. I think there's a group that wants to go to the folk event.
No. They want to hear about chum salmon first.
Okay.
So thank you very much, Ed. And that was a very interesting presentation. You kind of ended on some good news that there's a rebound in the thought process there, as long as we can keep the water cold. And now is the time for the Straight Science audience. And I know we have a caller, so we're going to throw that open to the caller. But before anybody jumps on with a question, be thinking of your question. And this is the time the Straight Science audience usually types in or shows with their little knowledge of however you do this, their appreciation for Ed Farley. It is never easy to be a public speaker, and I just want to give a shout out to Ed. That was a really great presentation. And just in case people weren't aware, wouldn't you know, always the good ones want to retire. So Ed Farley is retiring shortly, and so here he gives us this great information in such a great format. And as a fellow Alaskan, I'm grateful you speak to us all the way from Juneau. And I hope you have a great retirement. I am weeping that you're leaving the realm of the working, but I'm also a little jealous that you're out going to enjoy the new trail. So everybody write in a little something, and we'll start up with questions. The caller has the first go because it's never easy to be a caller on a Zoom call. So caller, if you want to unmute, and I think that's *6, or if you're having trouble with that, my cell phone, you can call me or text this number 907-434-1149, and I can we can do it that way and I can get it straight out here to Ed via the phone. Let's see if we've got questions. A lot of kudos.
Yeah, I'm here through November.
Okay, that's good to know. That's good to know. And also let us know who they've got trying to fill your shoes. That'll be a.
Yeah.
So I have a question. I'll go first. Oh, no, wait, there's Hio Eichen from UAF in the chat. And he says, question for Ed. Can you say more about the Japanese research? You mentioned that they did a couple of surveys in the Gulf of Alaska. Specifically, are they doing applied science that's tracking the fate of Japanese hatchery salmon?
Yes, they are. So they had more surveys in the past. Now they're focused primarily in the central Bering Sea, and they do have some survey tracks that are within the U.S. exclusive economic zone, and they do this in summer. What they're trying to understand is relative abundance and fitness and competition. So this is the Hoko Maru survey. And if you want to find out more information on those surveys, this is a survey they've been conducting annually. You can get that off the NPAFC website.
And I put that link to the North Pacific Anadromous Fish Commission is in the chat. I put the website up in there. And Hio says, great, thanks, Ed, very helpful. I've got a question, and that is, you had said about the jellyfish. The jellyfish in 2019 that caught my ear, they were so important, or I don't know if they're important. They were what the fish had to eat. That's what they were auguring in on. We have an increase of jellyfish, it seems, going on in the Bering Strait region waters. Is this corresponding, or would you know offhand, is this corresponding to the results they find in the northern Bering Sea with the, looking at the diet, what the fish are eating or the younger fish are eating on those surveys? Are they also increased in jellyfish in 2019 and beyond?
Yeah, so if you think about the juvenile life history stage for chum salmon in the northern Bering Sea, we do know that they were also eating cnidarians at a much higher proportion in this warmer period, which is very interesting to me because those cnidarians are offshore in winter, and they're feeding on. It seems like they like warm. Cnidarians do well in warm water, so they were also more abundant, and that's what these chum salmon were feeding on in the winter, in the warm year. Yeah, so there was a paper that came out by Dave Kimmel that looked at the increase in cnidarian during the warm years, because that paper examined the 20-year time series from our survey that we do in the northern Bering Sea, and there was an increase in cnidaria, and there was also an increase in chum salmon feeding on cnidaria. So that's shifted back. I don't know if that's the same now. I don't think the cnidaria are as abundant at this point.
Okay, maybe not as abundant and not a preferred food, would you say?
The chum salmon have a really weird stomach, and they will feed on jellies, and they do tend to feed on the lower, some of the prey items that aren't as good quality. However, they will feed on other things that are gelatinous too, that aren't as bad, like oikopleuron. That's another gelatinous critter, but they have a much higher energy density.
All right. Cool. Thank you so much. Charlie Lean, I see your hand up. Go ahead.
Yeah, Ed, thank you. It's very interesting. I have so many questions, but back in the 80s, 87 to be specific, we had a terrible chum run in Norton Sound and Kotzebue, and I was grasping at straws as the salmon manager to figure out why. And at that time there was an early breakup, and to me it seemed like the freshwater breakup didn't coincide with the saltwater breakup. The saltwater breakup occurred too early, so you could say that it was a warm water event or you could say that there was a disconnect between the two breakups. But anyway, I attributed it to be a disconnect, and I thought that survival had dropped to for chum salmon, and it was my working theory, and it seemed to work. So I just wondered if you had a comment on the productivity of the ocean. I mean, we've heard about the cold pool and things like that, but the early life history is close to shore. So anyway, comment.
Yeah, I don't know much about the nearshore ecology and what's happening there, but in the late summer, we do have 20 years of information that we can point back to in the northern Bering Sea that Dave Kimmel he's a colleague at Alaska Fisheries Science Center, just did a paper on zooplankton biomass and shifts in the species composition during our survey in warm and cool years. And it seems like there's ample prey. It's just what kind of prey, and that's the key question. And when it gets very warm up there in the northern Bering Sea, it seems, at least from the surveys that we did, that those prey items, specifically to chum, are a much poor quality, and there's more of them. There's more of that prey. So I don't know what happened in '87,
Yeah.
but in the 20 years of data that we've seen, it seems like things have been shifting a little bit, but it shifted a lot in 17 through 19 in terms of the types of prey, the amount of poor quality prey that was available. And I know that there was a lot of things that changed in that ecosystem, unusual mortality events, seabird die-offs, you name it. There was a lot of impacts.
So just to follow up, if I could, when we have less sea ice, we get less ice algae, and that seems like the productivity of the Bering Sea goes down, and that's another effect of the warm weather. So I was just throwing things around in my head, trying to cause and effect, and wondering about the lower productivity due to lack of ice.
Yeah, sea ice algae is really high in fat, and it's very good for especially the benthic, the bottom dwellers. So when you don't have sea ice, the sea ice is very interesting, because those phytoplankton, those plants that are underneath the sea ice, will grow in spring, even though there's ice there. And so you will have growth occur when the sea ice melts back. That extremely good quality, lower trophic level prey, which is the plant matter, is available then to the ecosystem. Most of that falls to the bottom, and that's why you've got a lot of very healthy crab and other clams and things like that. We've got walrus up there, everything feeding off the bottom. And when you don't have sea ice, then you don't have that high quality plant matter that's falling to the benthos, and then you usually, or bottom, and then you usually get other subarctic species that come in, which tend to be less quality, even at the base of the food web.
Thank you.
And it changes the way the system works. I mean, instead of having a system where things are going well on the bottom, it becomes a little more productive in the midwater, or and your surface. But then it just depends, what sort of quality of that base of the food web is there, and that changes then what's available to fish. Yeah.
And compound that with all the other issues with an ecosystem rolling into the southern ecosystem, rolling into the northern ecosystem, because they kind of can, and
yeah,
food, less high quality food, competition and predation. So thank you, Charlie. Anyone with a question? Rob Kaler in the chat writes, are chum salmon in the Bering Sea mostly wild, or is there some hatchery chum, Japan and Russia? Is there genetic work?
Yeah, so it depends on what the life history is. So if you're a juvenile, you're typically near shore or in the regions from where you just left fresh water. You're going to be there for a few months. There's not a lot of intermixing from Asian stocks, let's say, over into the northern Bering Sea and southern Bering Sea at the juvenile life stage. However, after the salmon have been out there for a year, as we note, there's your Western Alaska chum salmon, which are wild, move into the Bering Sea basin offshore, and you will find a lot of Asian fish there. Most of them, many of them come from Japan, which are hatchery. Some come from Russia. There's a big intermingling of all the stocks. So it depends on where they're at within their life history. But yeah, they will later on, after their first year at sea, they will move offshore and experience not only in the Gulf with other stocks, but in the Bering Sea.
Thanks, Ed. He writes. So write your questions in the chat or raise your hand, or just, if you're that caller still on, just blurt it out. It's okay to interrupt around here to a degree. I have another question for you, and that is the marine mammal portion. My ears went up, and you had this eDNA analysis, and I think that's a genetics. They take a sample of water and run it through the genetics machine, an analytical machine, and it's looking for sort of like leftover skin flakes and things like that from other animals in the area. And Dahl's porpoise came up. I know that, did you guys have observers for marine mammals on those ships? And I was wondering, if so, did that line up? Because I know you had the eDNA results, and I know you wouldn't be able to see a shark underwater very easily, but for the marine mammal portion, those Dahl's porpoise, I wondered if those eDNA results mirrored what people may or may not have been seeing on the ship. I just didn't know if you had observers.
We did not. And so that's a very good question. I do think that whenever we saw, it was dark, a lot.
Right.
As I recall.
Yeah, wintertime, right?
Even though we were still sampling at night, right? And so a lot of that eDNA probably came from nighttime sampling because we did 24-hour round-the-clock samples. So I don't recall myself seeing any marine mammals during the day.
You had the night shift.
Well, yeah, it was.
You had to sleep sometime during the day, maybe. All right. So your ultimate statement at the end was our chumps, we should see a rebound.
Yeah, everything's great and they're good condition now. Survival should be high.
Okay. Well, you give us good news. Good news.
I hope so.
Take it. Yeah, we do too. And we're
needed.
really grateful to hear that. Any other questions? Any questions from any of the communities or the island? Now is a good chance. Just unmute or put something in the chat. All right. Well, given that, so we'll conclude this. The next week, we've got 2 in the lineup for the next 2 weeks. So the next Straight Science after Ed will be Leah Valdez, I think, and Haley Schofield, and they're going to talk about their local work here on bumblebees, which I'm excited about because you never hear a bumblebee presentation, and it's going to be about Seward Peninsula bumblebees. So that's going to be on the 25th of April, followed on the 2nd of May. And Ed, you may be interested in this. This will be the UAF and Norton Sound Economic Development Corporation results on the Pacific halibut tagging work that they've done. And that'll be it's going to be Bering Sea, but I think they'll be talking about the Pacific halibut that were tagged just north of the island from Savunga and in that area. So we're pretty excited that we'll have both those presentations coming up in the next 2 weeks. With that, Ed, if you want to stay on for 1 second afterwards, and I see no other questions. Your talk was great. Thank you so much, everyone, for coming, and we'll see you on the 25th for bumblebees. Now, I should say, everyone go head down to the mini. There's a bluegrass, there's some bluegrass music going on, and that's pretty sweet for Nome on a Wednesday night. So it's at the mini. Get down there and you'll still catch it. They're just starting up. With that, thank you so much, everyone. We'll see you next time.